Welcome back to Synergistic Science. I'm Dr. Julianna Goelzer, and today we're going to be discussing the topic of DNA and chromosomes. All this information is coming from Essential Cell Biology, a textbook by Albert, which will be in the show notes. I highly recommend it if you're studying for the MCAT or taking a cell biology course. Right, so this topic comes from chapter 5. We're going to cover a couple of key ideas. The first is the structure of DNA, which will be the main focus of this lecture. And then the following one that will finish up chapter 5 will cover the structure of eukaryotic chromosomes and how that structure is regulated and maintained within the cell. And for a little humor to start us off, from the Amoeba Sisters, I have a comic that says, "Hey, Jerry, can you take a second shift today?" "Sorry, boss, I can only do it from 5 to 3. No one envisioned the job of the DNA polymerase shift manager. They didn't envy it, that's for sure. So with that, we're going to get into talking about different polymerases and how they maintain the structure of DNA. So DNA and chromosomes. During the 20th century, genetics emerged as a major scientific field. So to set the scene, think back to your Bio 1 days. You have Gregory Mendel messing with the genetics of pea plants. We had come to understand that genetics was inherited, could be passed down, but we didn't really understand what biomolecules were important, the structure of them, what role they really played in the phenotypic development of an organism. And so this brought about significant questions such as, what is the actual structure of DNA? We know now that it's a double helix. Thanks to Watson and Crick and some of their contemporaries. How are these genes passed down? How are they inherited or shared from parent to child? Is DNA or protein the actual blueprint of the cell? A lot of folks are arguing that protein or amino acids were actually the code to a given cell, given that there are 20 different amino acids, but now we are aware that even though there are only 4 distinct bases, DNA is the thing that holds the code to life. And then how does DNA actually fit into the nucleus? So how do we have many, many feet of DNA in a given cell, and how do we mash it into a tiny nucleus, but still be able to access that information? And so we've come a long way kind of to underscore this idea. Queen Elizabeth II recently passed in several years ago, and it's just interesting to see that we discovered the structure of DNA the same year that she ascended to the throne. So all of this information and many of these questions have been answered within one person's lifetime, which gives us hope, for the rest of the field and what could come about within our own lifetimes. So long before scientists understood the biochemical structure, they understood the idea of inherited traits. So again, thinking back to Gregory Mendel, the idea that we have, yellow pea plants and green pea plants, tall ones, short ones, even just like basic ideas that if you look at a child and see the parent, you see and begin to pick out distinct traits and know exactly where they came from. So we came from those ideas of seeing, okay, the phenotypic traits that could be seen passed down from generation to generation. Kind of the middle step is the idea that chromosomes could be stained and visible during mitosis. So you can see a karyotype here, lining up each of the chromosomes in a diploid organism. And so while we couldn't read the actual letters, you could begin to see traits of, okay, is it male or female? Does it have XY or XX chromosomes? How many should be present? Is there a trisomy when there shouldn't be? And kind of as an interesting side note, this karyotyping idea really took off around World War II. I'll have a future video on the idea of how we started karyotyping beetles. I know it's kind of strange, but that was actually in beetles and flies was where we discovered as a community sex chromosomes and the idea that the number of chromosomes and how large they are could help determine if an organism was male or female. I have a couple of images and a video here showing how you could actually stain and visualize the chromosome. So see here during mitosis, they're getting pulled to either pole of the two daughter cells. In interphase, you can see it's less distinct, I don't know, X-like sort of chromosomes. And over here, we have a video as well, where you can see they've labeled the chromosomes in red here, and you can see the cytoskeletal elements and the asters here in green that are actually helping pull apart the two daughter cells. So we have the big picture structure of, okay, we have linear chromosomes in a eukaryotic cell, but what does it actually look like biochemically? So this was hotly debated when you think about folks like Watson and Crick and Rosalind Franklin, all working to find the double helix that we see here today, that we would all immediately recognize as, oh, that's DNA. Around that time, there were a significant number of scientists that were working towards getting the crystal structure of DNA. And one of the other structures that was proposed was this triple helix over here on the left. This was proposed by Linus Pauling. And while it's not the main DNA structure that we know and love, you do actually find this triple helix in nature. So there are some occasional instances where DNA can unwind and actually form a triple helix. So he wasn't wrong. It just wasn't the main structure that is there the majority of the time. This is known as your typical like A DNA. You also have B DNA and Z DNA depending on how it's twisted. So to zoom in a little bit further, we have our DNA or deoxyribonucleic acid. It's double-stranded polynucleotide, meaning there are multiple nucleotides making up monomers here, making up this polymer. It's formed by two separate chains of covalently linked deoxyribonucleotide units. Okay, so if we look at the building blocks, it has a sugar phosphate. So the deoxyribo sugar in orange with a phosphate, it gets added to a base, either an A, a T, a C, or a G. And then those monomers can get added onto each other to form a linear strand. And then we can have two of those linear strands running anti-parallel, one going one way, one going the other, and then they will begin to twist to form the double helix. Each of the chains shown over here on the left is held together by hydrogen bonds, which are shown as the little like red hashes here. The nucleotides are composed of a nitrogen-containing base and a 5-carbon sugar, to which we'll add a phosphate. And again, the bases that we would see in DNA, keyword DNA, not RNA, we'll talk about that later. DNA are adenine, cytosine, guanine, and thymine. The nucleotides are going to be covalently linked together, and they're going to form a chain through the sugars and phosphates. This forms the backbone. So you're seeing it kind of here in yellow and blue here. The polarity is indicated with either a 3' or a 5', so 5 or 3 here. I really like this image from the Essential Cell Bio book because it numbers around or kind of connects what you should be learning in organic chemistry. So if you count around the sugar molecule here, this would be our first carbon, second carbon, third carbon that has the oxygen here, hence the 3' end. If we keep counting, we keep counting 4', 5', that CH2 group is what we would expect to find out here on the 5' end. So this image should help explain the numbering, so it doesn't always felt random to me of like, well, where are we getting the 3 and where are we getting the 5? It's all about counting around the sugar. So in the case you get a question of directionality, you can always kind of work backwards, use your organic chemistry skills and count around the sugar to find polarity. There are complementary bases in between the two strands that we were just looking at. C goes to G, T goes to A. The two polynucleotide chains in the DNA double helix are held together by hydrogen bonds here in red. So you'll notice there are 3 of them between C and G, 2 of them between 2 and A. And that would mean that the T and A grouping would be not as strong as the C and G because there's 3 bonds in that case as opposed to 2. This can be important if you're planning a PCR experiment. I would need more heat or more energy put into a reaction if there are more bonds or more C's and T's, whereas a lower temperature or less energy if it's an A and a T. Bases don't pair randomly. They should always be in C to G, T to A. There are some weird exceptions. If you're interested, you can go and look up I motifs and G quartets, where you can get weird foldings of these structures, but this is the typical one you would expect to see. Purines are the 2-ring structures, so that would be our G here and our A, where you notice 1 ring, 2 ring, 1 ring, 2 ring. Pyrimidines, I always think of a pirate having 1 gold ring. I know it's silly, okay, but those have 1 ring here, so T only has 1, C only has 1. Each purine-pyrimidine pair would then be known as a base pair. So this is just some key vocab I always underline so that hopefully you'd be able to get a multiple choice question correct on the MCAT. The 2 double strands will run anti-parallel. So thinking back here, we would have our 5' to 3'. Well, you'll notice on the other side, it's going, 3' to 5'. So they're running in opposite directions. The double helix contains roughly 10 base pairs per turn. So you should get 10 base pairs before you come around the circle. That is in the A type of DNA. It's not going to be in the triple helix that I had mentioned earlier. The twisting contributes to being an energetically favorable conformation, and it should be complementary. So the 2 molecular surfaces should fit together and form non-covalent bonds with each other like we saw in the previous slide. You also see on the right here that there's a major groove. So there's a wider gap here and a minor groove, a smaller gap here. These are important when we think about protein binding. So any of the transcription factors or polymerases that we'll see later, we'll be looking for these unique structures and the correct base pairs that are there as well. So based on this, how can the information for specifying an organism be carried in its chemical form? Well, we're looking at, okay, each of those letters would be holding that information, and it will be unique for a given species. Okay, more work has been going into this as we've gained better sequencing tools. Also, how can this information be accurately copied? We'll look more at that in chapter 6, but effectively having 2 strands bound to each other means that there is some redundancy and even more checks and balances to make sure that it is maintained over time. Big picture takeaway, the structure determines function. So as we study the DNA structure, we understand more fully how it can function, hold information, and specify an organism's heredity, basically. So the exact correspondence between these 4-letter nucleotides of DNA and the 20-letter amino acid alphabet gives us proteins and the genetic code. This gives us gene expression, or the process by which a gene can make a product that is useful to a cell. by directly synthesizing either a protein or an RNA molecule. Okay, so this could just be summed up in the term central dogma, which is the idea that you can take DNA that's double-stranded, make it into a single-stranded RNA, and then further produce a protein molecule from that, all with the starting molecule being DNA. Right, so next time we'll wrap up some more discussions on DNA structure and different chromosomes and how they're maintained within a eukaryotic organism. Thanks for watching.